Hey everybody, today I want to talk about genes and gene expression. So let's start with genes. What are genes? Genes are units of DNA that carry the instructions to build proteins. And these instructions are written as what we call a genetic sequence. So let's take a look at, we have these homologous chromosomes, this pair of homologous chromosomes. And let's just take a look at one of the pieces, the DNA from one of the pieces of those chromosomes. So that's what we're looking at here. And what you can see is that this DNA is divided into different genes. This is gene 1, and the DNA below that represents gene 2. So if we take a closer look at this gene, first of all, we can see this is a double-stranded DNA that we're looking at. There's a single strand, and there's a single strand. And what we're looking at here is the genetic sequence. And the genetic sequence, if you were to look at it and not know much about it, you'd say, okay, it's just made up of the letters C, A, G, and T, different orders of those letters. But it means something more than that. So my question to you is, what are we looking at when we're looking at those different letters? What is that gene sequence actually referring to? Well, each of these letters, C, A, G, and T, represents a different nucleotide. DNA is made up of 4 different nucleotides, and those nucleotides vary by the nitrogenous bases. Those bases can make it a cytosine, an adenine, a guanine, or a thymine. So when we're talking about a genetic sequence, we're talking about the order of those nucleotide bases. We're talking about the order of the nucleotide. And it's this order of the nucleotide, it's the order of these nucleotides that determines which protein is going to be built. When we talk about gene expression then, we're talking about making a protein from a particular genetic sequence. So let's take a look at this double-stranded DNA, and again, it's divided into genes. Here's what they're calling gene 1, gene 2, and then we're taking a closer look at gene number 3. And here's the DNA, and DNA is double-stranded, but we're only looking at one of these strands. And what we're looking at here is the gene sequence. It's the order of the nucleotide bases. It's the order of the nitrogenous bases. So to make a protein, there's a couple things that we have to do. First of all, we have to take that DNA, that genetic sequence, and we have to make a copy of it. And the copy that we make is called RNA, or more specifically, messenger RNA. And that occurs through a process called transcription. Then what we're going to do is we're going to take this mRNA copy and we're going to take this genetic sequence, and specifically we're going to look at these sequences of 3 bases that we call codons, and we're going to use that genetic sequence to make a protein, to make a polypeptide. And that occurs through a process called translation. If we look over here at the cell, we can see all of this occurring. So here's our DNA, our double-stranded DNA, and what we want to do is we want to take one of those genes and we want to express a protein from it. So the first thing that we do is we take that gene and we make an mRNA copy of it, and that occurs through transcription. And this occurs within the nucleus of the cell. Then what happens is that mRNA is going to move out of these nuclear pores into the cytoplasm. And then what we're going to do is we're going to take this mRNA, this copy of the gene, and we're going to read that sequence, and we're going to use it to make a polypeptide. We're going to use it to make a protein. And when I'm using the term polypeptide here, a polypeptide is basically just a linear sequence of amino acids. It's a protein in its linear sequence. Once it folds together and makes a three-dimensional structure, that's when it's actually called a protein, when it's in its functional state. But with regards to gene expression, you'll see the term polypeptide because we're just making this string of amino acids and it hasn't folded back on itself yet. So here we go. This figure represents transcription. And again, transcription is taking an mRNA molecule, it's making an mRNA molecule from a DNA sequence, from a gene sequence. And that's what's going on here. And I just want you to look at this for a second and kind of orient yourself. And just take a second and orient yourself. What do you see? Well, first of all, here is the mRNA that we're going to make. And to make that, we're reading the genetic sequence of a gene that's located on this DNA. And we'll get to this in a sec. But, there's two strands of DNA, and the top strand is called the coding strand, and the bottom strand is called the template. And when we make mRNA, we're going to use the genetic sequence of the gene from the template strand. So we have our mRNA molecule, we have the gene that's located on the template strand. What else is required? Well, we're going to need an enzyme to perform this activity. And this entire circular structure is the enzyme. And the enzyme that's going to do this is called RNA polymerase. So we need an enzyme in this case. And what else do we need? Well, we're going to build this mRNA molecule, and mRNA is a polymer, just like DNA. It's made up of individual nucleotides. And so if we're going to make this mRNA molecule, then we need free RNA nucleotides to use as building blocks to build them. And there's one other thing I want to point out here, and that has to do with this RNA polymerase. So what's going to happen is the RNA polymerase is going to do a couple things. First of all, it's going to unwind this double-stranded DNA. So we have to break the hydrogen bonds between the complementary base pairs, and the reason we can do that is so the polymerase can actually read the template DNA strand. So the RNA polymerase is going to unwind the double-stranded DNA. And the next thing that it's going to do is it's going to read this genetic sequence. We'll say the polymerase is moving this way. And so what it's going to do is it moves down, it's reading this genetic sequence, and it's building an mRNA molecule from it. So the mRNA polymerase is actually going to be responsible for synthesizing the mRNA. And the name tells us that. ACE, so we know it's an enzyme, and it's an enzyme that's going to make a polymer. What's the polymer that we're going to make? RNA. So this gives us a little more detail of the transcription process. So here we have our double-stranded DNA, and this is the gene that we want to look at, that we want to transcribe, turn into an mRNA molecule. So what's going to happen is an RNA polymerase comes along, and it's going to do a couple things. It's going to unwind the double-stranded DNA. And then what it's going to do is it's going to go down here on the template strand, and it's going to read the genetic sequence of the gene. And in response to that, it's going to add complementary base pairs to build an RNA molecule. And the nucleotides that we find in RNA are very similar to the ones that we find in DNA. In fact, in most cases, they're identical. So the nucleotides that make up RNA include guanine, which is the same as in DNA, cytosine, which we find in DNA, adenine that we find in DNA, except instead of thymine, which we find in DNA, we're going to add uracil. So RNA is made up of 3 of the same nucleotides as DNA, and the only difference is that instead of thymine, we have uracil. So let's look at how the RNA polymerase is going to build the RNA polymerase. So here it starts at this side, and it sees this T, and what's complementary to T? Well, A is. Then, well, maybe I should have started at this one. Let's start at this one here. So the RNA polymerase reads a C on the DNA, and the complementary base to that is an A. Then it reads a T, and the complementary base to that is an A. But now it gets to the adenine, and normally the complementary to adenine would be thymine, but we don't have thymine here. We have uracil. So instead of a thymine, it's going to add a uracil. And the RNA polymerase does this the entire way down the length of the gene. And as a result, it produces an mRNA transcript. And when you're looking at this, you know that we're looking at RNA because it has these U's. Okay, it has the A's, it has the G's, it has the C's, just like DNA, but the only difference here is that it has a U. A couple other details. This is our double-stranded DNA, and this is the region, this is the gene that we want to transcribe. And so when an RNA polymerase comes along, let's say the RNA polymerase gets the call and it's like, okay, we want to transcribe this gene. It needs to make its way to the gene. And what it does is it binds to a region upstream of the gene to be transcribed, and that region is called the promoter region. And so it binds to the promoter region. This is the RNA polymerase there. And that's when it begins doing its thing. It unwinds the double-stranded DNA, and then it reads the template strand, and it makes a new mRNA molecule from that. And this is another picture just showing the details of this process. So the RNA polymerase binds to the promoter. It starts unwinding the DNA, and it starts reading the gene and producing an RNA transcript. And then it just moves its way down, and it continues to do this for the entire length of the gene until it reaches the end of the gene, and then the RNA polymerase drops off, and you have your completed RNA transcript. And again, this is all occurring within the nucleus of the cell. Now, of course, it's more complicated than this. When you make a new mRNA molecule, let's go back here. So we have this mRNA molecule, and then we're going to shoot it off into the cytoplasm, and that's where it's going to be translated into a protein. But the mRNA molecule that's initially made isn't complete. It has to undergo some modifications, and that's what this is showing here. This is our initial mRNA, And there are regions in the mRNA that are going to be used to express the protein, and those are called exons. And we want to keep those exon regions. But then there's other regions that aren't involved in the sequencing of the gene, and those are called intron regions, and we don't want those. So what's going to happen is all of the introns are going to be spliced out, and so we only have exons. So we only have exons in this completed mRNA. And then there's a couple other regions at the 5' region. It forms this, what we call a cap, 5' cap. And at the 3' region, it forms this poly A tail, this string of adenines just going off the end. And there's a lot of research that goes into this, and finding out what these are for. And one of the things that these modifications may be for, especially things like the poly A tail and the 5' cap, is they may affect the regulation of gene expression. So just because we have an mRNA here, there's no guarantee that it's going to go and be translated into a protein. Okay, there's other factors at play that could prevent that mRNA from becoming a protein. And so this 5' cap and the poly A tail may be factors in regulating whether or not that occurs. All right, so what we've just done is we've taken the genetic sequence of a gene, the DNA sequence of a specific gene, and we just made an mRNA copy of it through a process called transcription. And now what we want to do is we want to take this mRNA sequence, and we want to make a protein from it. So we want to start taking this mRNA sequence, and we want to build a string of amino acids from it. And to know how this occurs, there's an important definition or important concept to introduce, and that's a codon. A codon is a series of 3 bases, a triplet of bases that codes for a specific amino acid. So if we look at the mRNA, we can divide this mRNA sequence into series of 3 codons, and each of those codons, or each of those series of 3 bases, is called a codon. So the first codon is U, U, U. And that codon is going to specify what specific amino acid needs to be added to the polypeptide. The next codon is G, G, G. And that's going to code for a different amino acid. It's going to tell you to add a different amino acid onto it. And then we have another codon, C, G, U. And that's going to specify another amino acid to be added on. And so it's the sequence of the mRNA, these codons, that are going to determine what protein we're going to build, what amino acids we're going to put onto this protein. And we can actually look at an mRNA sequence and determine what protein is going to be built. So here's our mRNA molecule, and parts of the mRNA molecule are not going to be translated. So translation is taking this mRNA molecule and making a protein from it. And so parts of the mRNA molecule, this part upstream here, is going to be untranslated. So that's not going to be involved in making the protein, or at least it's not the genetic sequence to make a protein. And instead, we're going to start at what we call a start codon called AUG. So translation starts at, again, what we call a start codon, and the sequence of the start codon is AUG. And then what we're going to do so our question is, we want to know which amino acid is going to be added onto the polypeptide chain using this AUG for instructions. And to figure that out, what amino acid is going to be added, you need to look at this codon table. And so AUG, well, you start over here on the first letter, and here's A. Then you go up top to the second letter, and here's U. So that takes us down here. And then you go to the third letter, and the third letter is G. So that directs us right there. So our first amino acid that we're going to add is going to be methionine. Let's do another one. AGC. What amino acid is going to be added to the chain when we see AGC? First of all, here's our A. There's our G. So we go G down into the A section. And then lastly, we have C. AGC, there we are. And so we're going to add serine. And what I'd like you to do is just go ahead and complete the sequence. Take the mRNA and make an appropriate polypeptide chain for it. So determine what amino acids are going to be added in each of these locations. And this is going to go all the way down to what we call the stop codon. Once the translational apparatus hits the stop codon, then we stop producing this protein. So what did we come up with? Well, hopefully this is what you came up with. There's your methionine, there's your serine, and then just on down the list. And here's another example for you to do that with. This is your double-stranded DNA. And here's the template strand down below. Here's the RNA polymerase. So what I'd like you to do is, using the genetic sequence of the template strand, I'd like you to tell me what the mRNA sequence is going to be. And then based on that, I'd like you to tell me what amino acid sequence is going to result from it. Okay, and what we're looking at here is translation. This is the translational apparatus. This is everything we need for the most part to perform translation, to take mRNA and turn it into a mRNA molecule. And there's a bunch of different kind of crazy stuff going on in here. So first of all, here's our mRNA molecule, and here you can see the genetic sequence for that. Another thing we need is what's called a ribosome. And this ribosome is kind of like the workbench where translation is going to occur. Another thing we need are these tRNA molecules. And attached to these tRNA molecules are going to be amino acids. So we need free amino acids to attach to these tRNA molecules as well. And basically what's going to happen here with translation is this mRNA molecule is going to go into the cytoplasm and it's going to hook up with this ribosome. It's going to be inserted into this ribosome, and the mRNA is going to move its way through the ribosome. And as it does, its genetic sequence is being read, and the appropriate amino acid chain is being built because of it. And so again, essentially what's happening here is the mRNA is moving through the ribosome, and as it's moving through the ribosome, the genetic sequence of the mRNA is being read, and there are corresponding amino acids being added to the chain because of it. And so now let's go right here, and the mRNA codon that we're dealing with is GGC. And what's going to happen, if you go look up GGC on that codon table, let's go ahead and do that. GGC. So the amino acid that we want to add is glycine. And so what happens is that here's what we call a tRNA, a transfer tRNA, and this tRNA has an anticodon region. It has the tRNA is basically going to read this genetic sequence and add the appropriate amino acid to the chain because of it. It's kind of a complicated process to try to explain, but let's see if we can do that here. So this is the tRNA molecule, and again, this is it's RNA. This is an example of an RNA structure in the body, and it's basically a linear RNA, but then it folds back onto itself because of this complementary base pairing, and it actually forms a complex three-dimensional molecule. And Meowsers. And at the top of the tRNA molecule, up here, this is where an amino acid is going to be attached. Okay. And then another important region that we have to consider on the tRNA is called the anticodon region. And man, this is difficult to explain, but here we have a GCC codon, and because of that, we want to add a glycine amino acid. But there's a bunch of tRNA molecules with different amino acids in them floating around the cell. So how are we going to know that this specific tRNA with the glycine around it needs to recognize that GCC codon? And the answer to that is on the tRNA molecule, there's an anticodon region. And what's going to happen is that the anticodon for the tRNA with glycine is going to be complementary to the mRNA codon. So we have C, G, Meowsers. And so the tRNA with an anticodon that's complementary to the codon is going to bind to that mRNA, and it's going to add the glycine. It's going to add the glycine to the protein chain. And now we're getting a little more, we're working ourselves more and more in detail here. So again, this is the translational apparatus, And what I want to point out here is that each ribosome has 3 tRNA binding sites. There's an E, P, and an A site. And E, a P, and an A site. And the P site is going to contain the tRNA with the growing polypeptide chain. This is the growing polypeptide chain that we're making. And then the incoming tRNA is going to bind into the A site. So again, here's the P site that has the tRNA with the growing polypeptide chain. We're going to have a new tRNA that comes into the A site, and the anticodon here is going to be complementary to the mRNA codon. And then what happens, so it has an amino acid on it. So now we're down to here. What's going to happen is this growing polypeptide chain is going to be transferred and added on to the new amino acid. And so now that's what we're showing here, is that this is our growing polypeptide chain, and this is the amino acid that we just added. Then what's going to happen is this empty tRNA is going to move to the E site, or the exit site, and then it's going to be removed from the ribosome. And then what's going to happen is this tRNA that just added a new amino acid is going to move into the P site right there. And then it just repeats itself. Then a new mRNA codon is read and a new tRNA comes in. And it does this over and over and over until it hits the stop codon. This is just a figure kind of summarizing everything that we just did. Here's our double-stranded DNA. And what we do is we read the template strand, and we're making an mRNA molecule because of that. There's the RNA polymerase. Then we're going to have some RNA processing that occurs. We're going to cut out the introns. We're going to add a poly A tail, a 5' cap. Then now that mature mRNA is going to move into the cytoplasm, and it's going to be taken up by the ribosome, and it's going to be shuffled through the ribosome. And as that happens, new codons are going to come through the different sites. And so here we have an AUG codon. So what happens is a tRNA with an anticodon complementary to that is going to bind to it, and it's going to add this new amino acid onto the polypeptide chain. This growing polypeptide chain is going to be transferred on top of that amino acid, and now we have a new amino acid in the polypeptide. Then this tRNA that's now empty moves to the E site and gets ejected. The tRNA that just entered in the A site goes in the P site, and then we move on and we read the next mRNA codon, and another tRNA comes in. And then in here, I just wanted to point out, because this talks about some material that we've already talked about. We talked about steroid hormones in the context of different types of lipids in the body. And because steroids are lipophilic, they're able to just diffuse inside the cells. Then they bind to their receptors inside the cells. And what do? They go inside the nucleus, and that hormone receptor complex binds to the DNA, and it stimulates transcription of a gene. That's how steroid hormones act, is they bind to a receptor which binds to the DNA, which stimulates gene expression. And thyroid hormone actually does the same thing. This is thyroxin T4. It diffuses inside the cell. It gets converted to the active form, T3. What does T3 do? It binds to a receptor that's located on the DNA, and that stimulates gene expression. It stimulates transcription of a gene. Now we're not quite done yet. So what happens here is here's the nucleus. So that's where we had transcription. And then that mRNA moves out of the nucleus, and these little dots are representing ribosomes, but these ribosomes are located on a structure within the cell called the rough endoplasmic reticulum. And so what happens here is that the proteins are synthesized in the ribosomes, but then they're taken up by the rough endoplasmic reticulum, and the rough endoplasmic reticulum takes these proteins and it encloses them into these vesicles. And then those vesicles with proteins move to another structure inside the cell called the Golgi apparatus. And this is where more protein modifications occur. And after it's done in the Golgi apparatus, we have a mature protein that's ready to do whatever it's going to do. And so one of the things that can happen to that protein is it gets taken up in a vesicle, and it gets taken to the plasma membrane, and it gets secreted out into the interstitial fluid. And it may travel through the blood and go somewhere else in the body. Another alternative is that protein is needed within the cell, in which case this secretory vessel may just open up within the cell, or maybe that mature protein is just secreted from the Golgi apparatus inside the cell itself. Actually, I should draw a protein like that, a three-dimensional protein. So the point I'm trying to make here is that the protein can either be secreted out of the cell if it's needed out of the cell, or it can stay within the cell and perform its function within the cell. And this is a good example of gene expression. Let's look at this picture. We have the two calf muscles, the gastrocnemius medial and lateral heads, and then those calf muscles are attached to the heel bone, or the calcaneus, by this tendon here. What's the name of that tendon? That's the Achilles tendon, or the calcaneal tendon. And tendons are super tough. And they're tough because they're made up of a tissue called dense regular connective tissue. And looking back to this picture, the reason it's so tough, the reason dense regular connective tissue is so tough, is because it's made of these collagen fibers. These collagen fibers are protein fibers, and they have high tensile strength. That means they resist stretching. They're super tough, which is what we want in a tendon. And then scattered, so these are the collagen fibers. And by the way, they all run in one direction, which means that they resist stretching in one direction. And then scattered throughout these collagen fibers are local resident cells that we call fibroblasts. And there's also cells in there called fibrocytes as well. So my question to you is, what if there's an injury to the Achilles tendon? Maybe your Achilles tendon stretches. It is tough, but if you stretch it enough, you can stretch these collagen fibers. So let's say that there's damage to these collagen fibers, and maybe even some of them completely rip. If that's the case, then you have to replace these collagen fibers. You have to replace these protein fibers. Well, how are we going to do that? Well, that's where these fibroblasts come in. These fibroblasts, within the nucleus of the fibroblasts, are all 46 chromosomes. And the entire genome, the entire protein encoding genome, is located in that nucleus. And guess what? On one of those chromosomes, or a pair of homologous chromosomes somewhere, there's the gene to make collagen fibers. And so these fibroblasts are going to be stimulated into gene expression, and what they're going to do is they're going to express the gene for the collagen fiber. They're going to make new collagen fibers. Well, then what? Do those collagen fibers stay within the cell? The transcription occurs in the nucleus. And then that mRNA moves into the cytoplasm and it gets translated into a protein in the cytoplasm. Then it goes through that endomembrane system. Okay, the rough endoplasmic reticulum and the Golgi apparatus, and finally it's ready. That collagen fiber is ready for use. And so what's going to happen? What are those fibroblasts going to do? Well, they're going to put them in vesicles and they're going to secrete them outside the cell into the interstitial fluid. So this is a good example of where the cell is going to make a protein, and instead of keeping it within the cell, it's going to send it outside of the cell. And just a side note, if you tear your tendon, do tendons heal quickly? And why or why not? And the answer to that is no. Tendons do not heal quickly. And the reason that's the case is because these collagen fibers are really dense. That's where they get the dents from. And there's just not a lot of room for blood vessels to penetrate that. So they have a poor blood supply. So it takes a long time because of that to, for a relatively long amount of time for the immune system to come in and for repair to happen and whatnot.